Okay, so for these lecture slides, we'll be looking at Chapter 4, Protein Structure and Function. So proteins are made up of amino acids. As this figure shows, we can see that there's a conserved region of the amino acids. And so that conserved region we see here in this gray box, those are going to be linked together through peptide bonds. And this is going to create this polypeptide backbone. So multiple amino acids coming together forms peptides. Long peptides would be proteins, but both peptides and proteins are made up of amino acids. So in this conserved area of the amino acids, we're going to have an amino or N terminus, and then we're going to have a carboxyl terminus or C terminus. So the easiest way to keep this in mind is N for the nitrogen, amino, C for carbon, carboxyl. Again, peptides bond holding these together. And then we're going to have these side chains. These side chains are unique to the amino acids. So we'll talk about the different amino acids shortly. And so if we start to recognize or memorize those side chains, we'd be able to tell the different amino acids that are present. So this process of linking amino acids together and forming a peptide bond requires the removal of water, so a condensation reaction. So if you think about condensation, water on the side of a cold glass, or condensation because you put your hand in front of your mouth and breathe, you're going to get water collected. Okay, and so by removing a water molecule from these two amino acids that are going to be coming together from their carboxyl and their amine ends, that will remove the water. We get the peptide bond forming between these two. So as I've already mentioned, if we take a generic amino acid, we're going to have that amine group, which is the nitrogen end, and the carboxyl group, which is on the carbon end. We're going to have carbon in the middle, hydrogen coming off of it, and then we're going to have our side group, or our radical group. Okay, and if we look at this side chain, this is how we'd be able to recognize our different amino acids. Okay, so we have 20 amino acids that are found in polypeptides. Of these 20, some of them are essential amino acids, which means we're not able to synthesize them, so we need to get them through our diet. Other amino acids are non-essential, so we're actually able to synthesize these. We have enzymes in order to make them, so we don't require on getting them through our diet. If we look at the 20 amino acids on this slide, we're looking at the non-polar amino acids. So we want to keep in mind with our non-polar amino acids, these would be hydrophobic. So they would be scared of water. So these would try to be buried into the interior of the cell, or into the interior of the folded protein. In panel A, what we're looking at are the non-polar aliphatic amino acids. In panel B, we're looking at the aromic amino acids. Okay, and so with the aromic amino acids, we can see how there's rings that are forming here. Where in panel A with the aliphatic amino acids, these don't form rings, these are chains. Okay, so keeping in mind those differences might be a way in which you can determine the difference between them. Okay. So for this slide, we're looking at the polar amino acids. So in panel C, we have polar amino acids but are neutral, so they don't have a charge to them. On the bottom panel, we have for D, these are the polar and acidic amino acids. So they're going to have a negative charge. And for E, we have our polar basic, so they're going to have a positive charge. Okay, so if the amino acids are polar and have a charge, these would be hydrophilic. So hydrophilic meaning that they like water, so they're drawn towards it. And so because of that charge, if you are acidic, so you have that negative charge, you would be drawn to the positive charge of hydrogen, and that could be in water. If you were basic, you have a positive charge, so you would be drawn towards the oxygen of the water, which would be negatively charged, right? So opposites are going to attract here. And so because you are polar and charged amino acid, you're going to be more likely to be the outside of the surface of the protein, so that you can interact with molecules like water and ions and such. Okay. So if we compare our polar amino acids to our non-polar amino acids, one of the things you'll start to notice is that we have these oxygen molecules out and available, or that we have these hydrogen. So again, we have a charge here. But if we were to compare those to the non-polar is going to have a lot of hydrogen and carbon, but we're not going to see the oxygen there in those R groups. So as we look at the amino acids, definitely keep in mind those side groups and what they look like, but also the characteristics might be an easier way to start to memorize your amino acid structures. So this is where I included this table. It's a figure, but it's a little bit more like a table, where it talks about the characteristics of the side group and whether it's charged, being polar, or whether it's non-polar. So in class on Monday, we will do an activity where we'll look at the amino acids, and you guys will get into groups if everything in technology works. And hopefully with that activity, that will help you to come up with ways in order to better learn, memorize the different amino acids. So now that we have an understanding of what the amino acids look like and that they have different characteristics, we can think about what are the consequences for changes in the DNA sequence, right? So if we change the DNA sequence, that potentially could change the amino acid sequence for a protein. And how would that change the protein structure? Right, so we do want to keep in mind that not all changes to the DNA sequence causes a change to the amino acid just because of how they're coded and that wiggle that you might have talked about in intro bio class that occurs in the coding. But if we have a change to the DNA sequence that changes the amino acid, it may change the amino acid to one that with a very similar characteristic, in which case we may not see much of a change. But in other cases, it could change the amino acid and it could have a very different characteristic, right? So if we went from a non-polar to a polar amino acid, that probably would change the protein. So it would change, because it changed the primary structure of the protein, it then would, and because the characteristics, how it folds is going to change, and that would end up affecting its tertiary structure and how it would interact with other proteins. And that could have a profound effect to the phenotype of the organism. This is being done into like a mouse model. So all those things can be studied. So next I'm going to play a short video looking at a case where we do have a change in the DNA sequence and it affects the protein structure.
Sickle cell anemia is a genetic disease that affects hemoglobin, the oxygen transport molecule in the blood. The disease gets its name from the shape of the red blood cells under low oxygen conditions. Some red blood cells become sickle-shaped, and these elongated cells get stuck in small blood vessels so that parts of the body don't get the oxygen they need. Sickle cell anemia is caused by a single letter change in the DNA. This in turn alters one of the amino acids in the hemoglobin protein. Valine sits in a position where glutamic acid should be. The valine makes the hemoglobin molecules stick together when oxygen tension is low, forming long fibers that distort the shape of the red blood cells, and this brings on an attack.
So we can see there how just a single nucleic acid being changed changes the DNA sequence and then changes the protein, the amino acid, and how then that protein can function. Okay, and so one of the things you can do with your pet proteins is to look, is there any kind of mutations that we know about? Okay, so not on the first worksheet that you have to pass in, but definitely might be something that is of interest because you're picking proteins that are of interest. So we talked about how we can have the interactions between the amino acids on those conserved areas, creating that peptide bond that holds them together. But we can also have interactions that occur between the side groups, or those R groups. So here in this figure, we see how we have the amino acid sequence, but as the protein starts to fold on itself, these side groups can interact with each other. And we can have different types of attractions that occur. So we can have electrostatic attraction that occurs because of the different charges. So we have a negatively charged and a positively charged, and so they're going to be attracted together. We can also have van der Waals attraction. So in this middle area here with the brown, we can see how these methyl groups of these kind of shorter side groups of these amino acids are interacting with each other. We can also have hydrogen bonds that are going to form between amino acids. So it's these interactions that are going to allow the protein to start to fold, and the protein will always fold in an energy minimized structure, so whatever the least resistance is. So we can see how we have this unfolded protein, we have these non-polar side chains, which are in the green, and then we have our other polar side chains in our unfolded polypeptide. As the protein, the polypeptide, begins to fold, those non-polar, those hydrophobic side chains are going to move towards the inside, and so that we'll have those polar ones on the outside, and those polar ones, because they may be charged, would be able to interact with other regions of the protein and also other proteins. Now, how do we know that proteins are using an energy minimized structure in its folding? So one way that an experiment that we could set up to look at this is by taking purified proteins from a cell. We expose it to high concentrations of urea. Urea starts to compete with those binding sites and actually ends up denaturing the protein. Now, if we then remove the urea, not adding energy, the protein will refold to its original conformation. Now, the reality is some proteins do need a little bit of help in order to fold. So if left on their own accord, they're going to fold incorrectly. So chaperone proteins located usually in the ER or in the cytosol would help to fold it so it's correctly folded and correctly folded for its function. So incorrectly folded proteins cannot function. So whatever type of protein they are, they would not be able to perform that task. So it needs to be correctly folded. And so maybe just a little bit of extra energy or twisting of the proteins needed, and this is where those chaperone proteins help out. Another way in which chaperone proteins can help out is they can actually create this kind of chamber. So the unfolded protein will move in there. Usually a cap protein or a chamber cap moves in, and this actually creates like a little, nest for it. And once it's isolated, it's able to actually fold correctly. Once the protein's folded, the cap protein or chamber cap comes off, and the correctly folded protein can leave that chaperone protein. So we want to keep in mind that proteins come in many different shapes and sizes. So, as you guys are selecting your pet proteins and researching them, you'll find that they range the whole gamut, right? So we can have very small proteins that in some cases are almost like peptides that may just function as a carrier, or we could have very large enzyme proteins that are going to be critical to the cell. We can have ones that have a very fibrous or kind of like string-like appearance, like we see here with collagen. And so just as this figure shows, they come in all different shapes and sizes, and that's what allows them to function and to interact and bind with other molecules and other proteins. So the way in which we can look at protein structure, there's different formats that we can look at it. So one way is to look at the backbone model. So this is just showing you that polypeptide backbone. So none of the side groups are in here. More popular is the ribbon model. So for your pet protein, I would imagine every one of you will be able to find a ribbon model of it. If you can't, let me know, because the ribbon model is very helpful when we start to look at some of the secondary structures that we'll talk about in a minute, but we can see how we can have those alpha helixes and beta sheets, and the ribbon model does a nice job of being able to see that. A wire model and space filling model fills in some of the side groups that are present, so we get a better idea of the interaction of those side groups that may be present. I would say that the space filling model is more common than the wire model if you're looking at like literature. So definitely in the immune system, when you're looking at antibodies and antigen, the space filling model is very helpful because you can start to see where the antigen, where this protein might interact with an antibody. Okay, and we'll talk more about antibodies shortly. So as I mentioned in that ribbon model, those are nice because you're able to see some of the secondary structures that are forming. And so the secondary structures that are common are the alpha helix and beta sheets. So in the top of this figure, we can see our alpha helix and how it's represented if we look at all of the different elements that are present. Versus a ribbon model. So again, the ribbon model is where you have that, it's almost like a curling ribbon that you might put like on a present. So it's going to be a nice curly cue. Beta sheets have this kind of back and forth. So I think of them as more like lasagna, like pasta sheets. So they're going to have more of a flat appearance, and they generally will run parallel in different directions. So when we have an alpha helix, alpha helixes are good at being transmembrane. So if you notice that your protein has an area that the alpha helix is kind of gapped between, is connecting two different parts of your protein, that could be because it's a transmembrane protein. And so this hydrophobic region here, it doesn't want to be around the water, and so that biphospholipid layer creates kind of like a place for it to go. Alpha helixes can also twist together to create more complex structures. So for beta sheets, there's going to be two different flavors, if you will. So anti-parallel, as I mentioned, is probably the more common one because this allows for short turns in the protein. But there's also parallel beta sheets, so they're going to run in the same direction. In order to have a parallel beta sheet, you need to have a long turn, right? So the turn for an anti-parallel beta sheet may be only several amino acids long, where for a parallel beta sheet, this would have to be significantly longer than with anti-parallel. So many proteins are composed of separate functional domains or regions. So in this figure, we see the secondary structure where we have the alpha helix and the beta sheets. Those are going to come together to form a single protein domain. But we can see how on the right side of this figure, this protein actually has two different domains that are connected together through that amino acid sequence. And so those domains coming together, this whole protein folding into its final structure, would be its tertiary structure. So again, some proteins may have domains as part of their tertiary structure. We'll make a distinction between quaternary structure. Quaternary structure is where we have subunits coming together. Okay, so if these were not linked together, that's where that would be quaternary, because we'd have two subunits, but we can see how there's a connection between there. So they have domains, tertiary, subunits, quaternary. So the benefit of having these connected and having these separate domains is that there's more flexibility in it. So it's kind of free to adopt some novel conformations. So from like an active form to an inactive form, or maybe being able to bind a substrate in there. Okay. So here we're looking at a protein that has a quaternary structure. So we have these alpha subunits that come together and our beta subunits, and plus we have additional proteins that are going to come in to allow for it to function. So this molecule here is hemoglobin. And so that red that we're seeing is the heme group, and that is what's going to be able to bind to iron. Okay. And that iron then allows us to bind oxygen. Okay. But both that beta and alpha are hemoglobin molecules. This whole structure would be the quaternary structure, and this is where the protein is going to be functional. So without this heme group, the protein is not functional. Proteins can also bind to subunits and polymerize. So we can have either we can have free subunits, but maybe they would bind to another subunit and then they would form a dimer. If we had multiple subunits coming together, we could, again, we're forming a polymer, and this could either have a helical shape to it, or maybe we're going to form a ring. So molecules like actin will form a polymer, and that allows it to function. So its tertiary structure, its quaternary structure is this binding together these different subunits. So one way in which proteins are able to stabilize themselves, because keep in mind many proteins are moving around different parts of the cell, or moving outside the cell and inside the cell, and they're going to be exposed to many different types of environments, and they want to try to keep themselves folded. And so one of the ways they do this is by disulfur bonds. So these disulfur bonds that we're seeing in the figure here, we have two sulfurs that are in the side groups, and they're going to then interact with each other. And again, this helps to stabilize the protein. So disulfur bonds are going to be stronger than those other interactions that we were talking about, like hydrogen bonds. Proteins can also bind other molecules, and they do this in a highly selective manner because those exposed side groups can only bind certain molecules. Okay, so what we're seeing here are the side groups that are depicted by these yellow, red, and blue dots. And so they're going to be limited in what they can bind, so that binding site becomes very specific. And that's how your receptors or like antibodies would work is because of the side groups available to that receptor, like insulin receptor, it's only going to be able to bind insulin. Just like those binding sites, like on an enzyme, would only be able to bind specific substrates too. So, as an immunologist, I like to talk about antibodies. And so here we have this picture of an antibody, and it's got a lot of the different features. It's got different domains. So we have this light chain that's got 2 different domains to it. It's complexing with this heavy chain. We have sulfur bonds forming to stabilize it. And then we also have this antigen binding site where we have, for each antibody, different type of antibody, we would have different side groups for the amino acids. And because of those different side groups, it would allow us to bind specific antigen. So the antibody that may recognize like E. coli is not going to be the same antibody that would recognize strep. It wouldn't be the same antibody that would recognize malaria. It wouldn't be the same antibody that you would use in doing a Western blot for actin, right? So each antibody would have specific amino acids that would be present in that area of the antigen binding site, and those side groups would then determine what it would be able to actually bind. So as I was mentioned, this figure is just kind of giving you a visual of what I was just talking about. Antibodies are going to bind other molecules in a highly selective manner. And so the antibodies that might recognize foreign materials like pollen are going to be different. So we don't tend to get the cross-linking between different antigens happening. Sometimes it can, but usually that would mean that those molecules are the same on the different sources. So next we're going to talk about some of the different ways in which proteins can be regulated or controlled. This is really important when we're looking at enzymes, right? Enzymes are proteins that are going to catalyze reactions. So we want to make sure that those reactions are only occurring when they're needed. So, if we have too much product, we don't want to make more product. Or if we don't have enough substrate, we can't make product. So again, we want to be able to control those. Also with, looking at gene regulation, we want to make sure that if we're turning a gene on, we're only turning the gene on when we're going to need it, whether it's the stage of the development, right? So sometimes the product that is formed from a reaction will help to regulate. So there might be a binding site for regulation as also a binding site for the substrate. So what we're looking at here is an enzyme that is going to catalyze a reaction for sugar. So it's going to bind like glucose. But the presence of ADP is needed in order for that binding and that closing of the enzyme to catalyze the reaction, to catalyze the next step. Now, keep in mind that ADP cycles from ADP to ATP, so diphosphate, triphosphate. So if we think about, this is where, if I had a whiteboard, this would be so much easier. If we think about in a cell, if we have high amounts of ATP, we're going to have low amounts of ADP. Okay, so if the cell has converted the sugar, the glucose, into energy, into ATP, we're going to have high amounts of ATP present. We're not going to have a lot of ADP present, right? So that's going to be similar to what we're seeing in panel B, right? So there's not a lot of ADP there, and so the enzyme's not really able to bind the substrate, the sugar. It's not able to kind of lock that in. to catalyze the reaction. Now, when we've used up the ATP in the cell, it's going to convert to ADP. So we're going to end up having high amounts of ADP, but low amounts of ATP. So we need to make more ATP. So that scenario is what we're looking at in panel C. So now that the ATP has been used up, it's been converted to ADP because that phosphate's been removed to make maybe peptide bonds. You're going to have more ADP. Because that ADP is binding to the enzyme, that allows that enzyme to close itself on the glucose molecule on the sugar, and allows it to catalyze that to start the process of converting the glucose into energy. Okay, so having that binding site creates a regulation, okay, where when we don't have as much product, and in this case the product would be ATP, we're able to then bind the ADP so that we can make more. So again, this binding of cofactors is one way in which proteins can be regulated. Okay, so another way in which proteins can be regulated is through post-translational modifications. So this can help control the protein's function, but also the location in which it's found in. So in this figure, we're looking at the protein P53, which is a cell cycling protein, and knows to play a role in some cancers. So what we're seeing on here is kind of a map of the different post-translational modifications. So we can see that we have phosphorylation that can occur, acetylation, ubiquination. So all of these different post-translational modifications, some of them can happen in the ER. Other post-translational modifications can happen when it's in the cytosol or outside the cell. Phosphorylation, generally we think of this as like an activation of the cell. So you phosphorylate it, and this causes a signaling cascade, so it kind of activates the next protein. So when we talk about cell signaling later on in the semester, we'll talk about phosphorylation. Ubiquination usually signals the protein to be degraded. So basically its life is over, and so it will go to the proteasomes, get degraded, and amino acids will be recycled into synthesizing new. There are other post-translational modifications, so methylation. There's also a post-translational modification called citrinylation, not studied quite as much, but some of these different post-translational modifications can be significant enough that the immune system can recognize when there's a modification versus when there's not a modification, and it may respond to modified or unmodified proteins, whichever one's not the normal, okay? So just kind of something to keep in the back of the mind if you go on and study some immunology, okay? So that's going to conclude the lecture for these slides for Chapter 4. As I had mentioned on my announcement, we will be doing an activity in class on Monday looking at kind of helping you to learn more about the amino acids, because I think that's the piece that probably will be most difficult for students with this material, is that kind of getting memorizing. So I highly recommend using this time to, maybe print out those slides that have the individual amino acids on, and making flashcards to start. But hopefully our activity on Monday will also be helpful too. Okay, so thank you very much.